Hello everyone, I am Sita Lakshmi from 2nd MSc Biology. Today we are discussing about the topic processing of tRNA and rRNA. tRNA processing. A immature tRNA or precursor tRNA is converted into mature tRNA. There are several events that take place. This process is known as tRNA processing. Okay, it is occur in both prokaryotic and eukaryote. The mature tRNAs consist of 80 to 90 nucleotides, but the precursors, that means the first formed tRNA. The precursors tRNA is much longer that contain many nucleotides, and the unwanted nucleotides become cleaved off with the help of enzyme RNase D, E, F, and P. Nucleotides are removed from 5' and 3' end. The cleavage at 5' end is catalyzed by the enzyme RNase P. Look at the image. There is a 5' end and 3' end. And the 5' end and 3' end cleaved by corresponding enzyme. For an example, 5' end is removed by RNase P. So the removal is take place, then adding a CCA nucleotide on the 3' end and changing some nucleotides for forming abnormal bases, it becomes mature tRNA. The cleavage at the 3' end is catalyzed by conventional protein RNAs and at the CCA terminus. Also mentioned the CCA terminus is added on the 3' end, and this is a post-transcriptional processing step. Some bases are modified at the catalytic position in the tRNA molecule. These are the dihydrouridine, ribothymidine, pseudouridine, inosine, and methyl glucosine. And methyl guanosine. And some pre-tRNA introns are removed by splicing. Okay, splicing is also involved in here. And an endonuclease cleaves the pre-tRNA at a splice site to excise the intron, and followed by the joining of exon to form a mature tRNA molecule. And first the end, the 5' 3' end is removed, then with the help of corresponding enzyme, then add the CCA terminal on the 3' end, then the rearrangement of the nucleotide, then splicing is occur, and finally we get the mature tRNA. This process is known as tRNA processing. And the second one is rRNA processing. rRNA, which means ribosomal RNA. The ribosomal RNA is different in prokaryotic and eukaryotic. The rRNA processing is different in both prokaryote and eukaryote. In the case of prokaryote, that contain 3 rRNAs, that is 16S, 23S, and 5S, which is formed by the cleavage of pre-rRNA transcript. And the initial cleavage of the bacterial pre-RNA yields separate precursors for the 3 individual rRNA. Thus, rRNA further processed by secondary cleavage to the final product gives. So, the primary cleavage is take place and the secondary cleavage is take place and get final product in prokaryote. But in the case of eukaryote, there are 4 rRNAs. So, the one of 55 rRNA is transcribed by separate gene, and others are derived from a common pre-rRNA. And the only rRNA that is not processed extensively in 55 rRNA in eukaryote. which is transcribed from a separate gene. And in eukaryotic cell, the pre-rRNA is first cleaved at the adjacent site to the 5.85 rRNA on its 5' side to separate the precursors that contain 16S and 28.5 plus 5.85 rRNAs respectively. Further, the cleavage then convert this to the real final product, and the 5.85 and rRNA become hydrogen bonded to the that are the main difference. The 5.85 rRNA is hydrogen bonded to the 285 molecule. In addition, this cleavage rRNA processing involve in addition the methyl groups of to the bases, ensure moieties of the specific nucleotide, and conversion of some uridine to spirouridine. Okay, there is a prokaryotic pre-rRNA is converted into mature rRNA. There is 16S and 23S, then 5S are corresponding to form to remove the unwanted by splicing. But in the case of eukaryote, the rRNA is converted to mature rRNA. There is a different or major highlight, which means the 5.85 and through 285 molecule have become hydrogen bonded. That are the main difference in prokaryotic and eukaryotic. And the immature pre-rRNA is converted into mature rRNA for further processing. That means transcription and translation. Okay, whatever that become mature and it become functional. That process is known as rRNA processing. Thank you.